Good day, grade 12 learners. Welcome to today's Life Sciences lesson. My name is Nonhlanhla Mtshali. This lesson is brought to you by the Department of Education in collaboration with Saibono Discovery Center. Today's lesson is on Life Sciences Paper 2, the revision in preparation for Monday, which is the 18th of September. So we're going to be looking at highlighting the things that are important, things that you should know before you go into that exam room on Monday. And as usual, we always start with the exam guideline. So the content that will be in your paper on Monday, if you don't know by now, it is the DNA code of life, meiosis, genetics and inheritance, and evolution. So I would say that the advantage with this paper is that there's fewer topics compared to paper 1. That is why we expect all of you to do very well in this paper. And again, we also look at the weighting. As you can see, evolution weighs 54 marks, genetics and inheritance is 48 marks, and then the other two topics are 27 and 21. So these are the topics that you need to prepare on for next week. And then again, we are going to touch on the resources. Remember, when you study, there are things that you must make sure that you have in preparation for your papers. So we have been giving the study guides and the seed material. You do have all the past exam question papers by now. Your teachers have shared the trial exam papers from Gauteng and from the other provinces. You have your exam guideline, you have the Mind the Gap, and then we have the websites that are very important that you need to consult, especially if you don't have all the past question papers. Then you can go to the Department of Education's website, which is www.education.gov.za. For the lessons that we are doing now and the ones that we did previously, you can go to www.pocketschool.co.za, and then we have this Google Drive link which has all these resources. So all the resources that are written here, you can find them in this Google Drive link. So then you can download and work on all the past question papers that you need to work on. Okay, then let's look at the topics. We said it's DNA and RNA, it's meiosis, genetics and inheritance, and then we said it's also evolution. So when you look at DNA and RNA, what is it that you must know, or what is it that you need to focus on? With the DNA and the RNA, remember, it is important that you do know the structure and what the location and the function. So structure of the DNA and the RNA, where are they found in the cell, and what are the functions of the two. Also, with the DNA, you must know what DNA replication is and why is it important. Remember, when we talk about DNA replication, we're talking about the process where the DNA makes copies of itself so that it can be shared between the resulting daughter cells during cell division, so that the daughter cells have got the exact same copies as the original cell. Also, what is important here is the DNA profiling. What is it that you need to know in DNA profiling? The uses, the interpretation of the DNA profiles. And then the other topic is the protein synthesis. What is protein synthesis? The process by which proteins are made in a cell of an organism to form enzymes, hormones, and any new structures for what? For the cell. And remember, with protein synthesis, the focus is on the two steps, the first one being transcription and the second one being translation. So the possible questions, when you look at DNA and RNA, the possible questions that you can come across, you will definitely find questions that are based on terminology. These are all your definitions, the ones that we've been telling you from beginning of the year, that if you come across a new term, you write it at the back of your book or you write it in front, wherever you write it. But by now, we are expecting that you have a vocab of all the life sciences terms from beginning of the year up until now. And then the interpretation of diagrams. So you'll be given a diagram, you can be asked questions based on the diagram. Tabulation, there can be questions where you'll be asked to tabulate the differences between the DNA and the RNA, or the differences between the DNA replication and DNA, transcription in protein synthesis. You can be asked to describe the process of DNA replication or describe protein synthesis. There could be calculations. Some of you if you've done as many past question papers as you can, you've seen that you can be asked to calculate the number of nucleotides. You can be asked to calculate the number of guanine molecules that will be in this molecule of the DNA or of the RNA. So then with the DNA and the RNA, if you're looking at the structures and the differences at the same time, here in front of us, we've got the diagram that shows the DNA and the diagram that shows the RNA. So what are the differences that you are looking at? You start with the sugar. So with the sugar, the DNA sugar is called the deoxyribose, whereas the RNA sugar is called the ribose. Hence the name deoxyribonucleic acid and ribonucleic acid. And then the DNA is a double-stranded molecule, which is why we call it double helix, because it is double-stranded, and it is a double-stranded molecule that is what is twisted, hence the double helix structure. And while the RNA are not double-stranded molecules, but they are single-stranded, as you can see in the diagram. So another thing with the DNA is that these two strands are held together by the hydrogen bonds. What you see here in between is what we call the hydrogen bonds. These are the things that keep the two strands together. And then the other difference between the two is that your DNA molecules are very long. These are very long molecules, whereas the RNA molecules are shorter. Both of them are made up of the building blocks that are called the nucleotides. So when you're asked what are the building blocks of the nucleic acids, those building blocks, we call them the nucleotides. So here we have the diagram. This is the DNA. This is how it looks like when it's uncoiled. And then this is what it is, the RNA, which shows that it is only made up of one strand. And then what are the nucleotides? This whole thing here represents the nucleotide. And again, if we look at the DNA molecule, this whole thing here is what we called the nucleotide, the building block. So each nucleotide is made up of the what? It is made up of the nitrogenous base. Remember, the your nitrogenous base in this case is the what? It can be, if it's the RNA, it can be the uracil, we have guanine, we have cytosine, and we also have the what? The adenine. So this is the nitrogenous base. Then it is also made up of the sugar molecule. So there is a sugar there. And then the third thing that makes up the nucleotide is what we call the phosphate. So these are the three things that make up the nucleotide. Same thing with the DNA. The nucleotide is made up of three things: the phosphate, the sugar molecule, and the nitrogenous base. So the other difference between the two is that the DNA has a nitrogenous base called thymine, which we represent it by letter T. It does not have what? The uracil, but it has the thymine, and then it has all the other ones, your adenine, your guanine, and the what? And the cytosine. So those are the other differences between the two. So you should earn all the marks here if there's a question in the paper that says differentiate between the DNA and the RNA. And then while you are studying, while you are revising your DNA, do not forget to look into DNA replication. We did say earlier that DNA replication is when the DNA makes the exact copy of itself. So here on the right hand side, we have the diagram that shows exactly what happens during DNA replication. This is the schematic representation. We've got 5 steps here. Step number 1, where we say it unwinds. Step number 2, which is unzipping. Step number 4, where we talk about the templates. Step number 3, and then we have 4, where we talk about the complementary strands that are formed afterwards, and then at the end there will be 2 identical molecules. So with step number 1, remember we said the DNA is a double helix structure, so it is winded up like this. So with step number 1, this double helix structure, it unwinds and then it looks like a ladder. Those of you who know what a ladder is, it looks like that. So here, that is step number 1, it unwinds, and then after unwinding, what happens is the bonds, remember we said the 2 strands are held together by bonds. So the bonds between the 2 strands are going to break, and the two strands will do what? Will unzip. Just like when you open the zip. Picture when you open the zip, it's the bonds that are breaking, and then the zip, the two pieces that make up the zip, they move apart. So that is when the two strands separate. And then with the third step is that now each strand, so we started with it being double helix, it unwind, and then we've got two strands, and then the bonds between the two strands separate. Then we have now the two strand. We have strand this side, we have the strand this side. So each of these strands, they do what? They serve as a template. See, we've highlighted all the important names for you. Each original DNA strand serves as a template on which its complement is built. So we've got the old strand that serves as a template, and then we have the new strands that are forming inside. Those are the complements. And then with number 4, the free nucleotides then will build onto the DNA strand, on each original strand, two strands, by attaching to the what? To the complementary nitrogenous bases. So remember, when you were told about the functions of the nucleus, or what is found in the nucleus and the nucleoplasm, we say that in the nucleoplasm you will find the free floating nucleotides, whether they are RNA nucleotides or free DNA nucleotides, they are just there. So if replication takes place, then those free floating nucleotides will then move to attach themselves on the original strand to form the two identical DNA molecules, and that is step number 5. But here, what is important is that you do not forget to mention the pairing that is complementary, that when we talk about the DNA, A always pair with what? With T. So adenine always pairs with thymine, and then cytosine always pairs with what? With guanine. This is the complementary base pairs that we are talking about. And then at the end, we have two identical what? DNA molecules that are formed. And note, we are not saying the DNA strands, but we are saying the DNA molecule. So this is the DNA replication. Here, if a question like this comes up on Monday, expect to get something like 5 to 6 marks. And this is something that you can memorize and know off by heart. Then we say each molecule then consists of one original strand and the old strand. So in the new DNA molecules that are formed, we have the old and the new strand that was formed by the free floating nucleotides. Then there's two processes. Then the second process that you need to know under DNA and RNA is the one which results in the formation of what? Of proteins, which is called protein synthesis. Protein synthesis occurs in 2 stages. There is stage number 1 that is called transcription, and stage number 2, which is called translation. Transcription takes place in the nucleus. Those of you who are not sure what this diagram is, this part represents the nucleus. So this is where stage number 1 takes place. And then stage number 2 takes place where? In the ribosomes. Where exactly? In the cytoplasm. So with the transcription, we're saying, remember, the proteins are formed. So what is the role of the DNA in protein synthesis? The role of the DNA is to provide the code. So the DNA provides the code on how the proteins are going to be built, or on how the amino acids will be aligned, so that at the end we have this particular protein. So with DNA, with transcription, we're looking at the double helix DNA again, same thing. Remember, if you have been studying, you know what I'm talking about. Transcription, DNA, it unwinds, and then after unwinding, what happens? The hydrogen bonds between the nitrogenous bases, they break, and the strands, it unzips. Be careful with this section. Remember, the bonds are the ones that break first before the strands can unzip. We do not start with unzip and then we say the bonds break. That's how you lose marks. So it unzips first, the bonds break first, and then the strands unzip, or they separate. So here, note that one DNA strand, only one DNA strand here will act as a what? As a template for the construction of the what? Of the mRNA. So here's our first and second difference. The first difference is that between this and replication is that one DNA strand acts as a template, and the construction is what? We are constructing the mRNA but not a DNA molecule. And then here another difference is that the free RNA nucleotides from the nucleoplasm are the ones that form the mRNA. So the mRNA complements the DNA in the what? In the nucleus. So remember that side I said A complements T and C complements what? G. Here we have A which will complement what now? U. Because remember, in the RNA, we do not have thymine, but we have uracil. So the complementary base pairs will be A complementing U and C that complements what? G. Then the mRNA complements the molecule, and that's the mRNA that is formed. So this is what is happening here, and then this is the mRNA now that has been formed. Once the mRNA is formed, it leaves and then it goes where? To the ribosomes. It is now carrying the code. So DNA has provided the code was transcribed, and it is carried by the mRNA. mRNA is now going to tell the tRNA in the ribosomes, which, okay, this is how you bring in the amino acids. This is the order now. So we are going to follow this order to make a protein. So then with translation in the ribosomes, so the coded mRNA, after it has left, it goes through the nuclear pore and it goes to the ribosomes. Then the tRNA will start picking up the amino acids, and they will make sure that these amino acids that are brought, or the tRNA, it complements the what? The mRNA on the ribosomes. The same complementary that we are talking about here, where this pairs with this and this pairs with that, A with U and C with G. It's the same order that we are talking about. And in doing so, the amino acids are already lined up. So here we have the tRNA and they are carrying amino acids. So they are already lined up, and as they sit next to each other, they then bond, and they are joined by these bonds, which you call the peptide bonds, and a protein is what is formed. So this process is very important. Usually, a lot of times, the questions that you will come across, they will ask you about transcription, but there are also question papers where translation is. So you are safe if you know both processes. Again, this is the process that you can just memorize, know it off by heart, so that you are able to answer when you get there on the paper. So the only challenge that we are picking up here is that as soon as you get the table, then that's where most of the learners struggle, because now you must work. Remember, sometimes you will be given a table of the base triplets of the DNA, and you are asked to provide the codons or the anticodons of the tRNA. So you must just know that if you have that table, I'm going to draw 3 lines, 3 columns like this. We have your DNA. Let's say in the first one we have the DNA base triplets, and then the second column is the mRNA, and then the last column will be your tRNA. We always tell learners that what is in this column, the last column and the first column, it is exactly the same. The only difference is that where there's a T, the T will be replaced by a what? By a U. The only one that is the different is the mRNA. For example, let's take the DNA, the mRNA that we are provided here. We have UCA, which is the first 3 over here. We also have GGC, which is the second codon, and then we have the last one, which is what? ACA. So if you are asked to give in the tRNA, your tRNA here will be AGU, because it is the opposite of this and it. So A pairs, U pairs with A, C pairs with G, and A pairs with U. Then for the second one, because it's GGC, it will be CCG. Then the last one will be UGU. So now, If the question says you are given the tRNA anticodons and work out the base triplets, I said this is exactly the same as what? As this one, except we don't have a what? AU on it. So the first two, AG, will be exactly the same. It will be AG, but we do not have a what? AU on it. So what is going to replace a U? It's going to be a what? A T. So we have the AGT. Again, with your GGC, it is exactly the same as this, so we will have the CCG again here, because we started with the DNA that provided a code which is CCG, and then the mRNA is GGC, then the tRNA is CCG. So that is how you work it out. Your first column and the third column are more or less the same, except where there's a T, there's a U, or where there's a there's a T, and then the one in the middle is the one that is completely different. Okay, now let's look at the differences between the replication and transcription, which is what you've already said. We said with the protein DNA replication, both strands act as a template. Transcription, only one strand acts as a template. DNA replication, free DNA nucleotides, RNA, free RNA nucleotides. And then the complementary base pairs are A pairs with T, G pairs with C, whereas here A pairs with U, again G pairs with what? With C. And then at the end we have two identical molecules of the DNA that are formed, whereas under transcription, mRNA molecule is formed. So these are questions that you can get. You can be asked differences between RNA and DNA, or differences between the DNA replication and the transcription. Okay, then under DNA, we also have what is called DNA profiling. So with the DNA profiling, remember a DNA profile, it is just a pattern of what? Of the black bars that represents the DNA fragments of what? Of a person. And it is said that we have a unique, each and every individual has his or her unique DNA profile, except for the what? For the identical twins. So this is the scientific process, the science that is involved here, so that these are put on a slide like this, so that you can see the patterns of the black bars representing these DNA fragments. So here what you must know is the importance. Why is it important? Where is it used, DNA profiling? So DNA profiling is used in paternity testing to find the parent who's the father, to trace missing persons, to identify genetic disorders, to identify criminals in what? In forensic investigations, to establish family relations, and to match tissues for what? For organ transplant. So there's many uses of the DNA profiling. So here your teachers have told you that each time you come across a question on DNA profiling, you need a ruler with you, because sometimes you will be asked, there's a mother and the father, and the child, but then the question is, who is the father between male 1 and male 2? So how are you going to study this? You need to have your ruler, so that you put it on each bar to see whether the bars match across from the mother, the child, male 1 and male 2. Remember, if the male is the father, you'll have that there will be bars of the child that match with the father, and then the ones that are remaining will match with what? With the mother. So that is how you study your DNA profile. You take your ruler and then you underline. If you can just at the glance and you look at this, you can already see that this child has this band which is found here, and it is also found where in male number 1, and male number 2 does not have. Again, if you look at band number 3, there it is, and same band is there in male number 1, which is also not found in male 2. With again 3, this one and this one, they do match. And then if you further go down, this one and this one also, they match. So we can conclude that in this DNA profile, this child, the father of this child, is male number 1, because more DNA bands on this male 1 matches the DNA bands of what? Of a child. And that is how you answer. You don't just say because the DNA profile of the father matches the DNA profile of the mother. You refer to the fragments, the bands, that the bands in the male 1, they match the bands, the DNA bands in the child's profile. So then that's how we That's what you should know when we talk about the DNA profiling. So if we wrap up DNA and RNA, what is it that you need to remember? The history of the discoverer. Remember, this is very important. You will find this question in question 1.1, 1.2, and 1.3. Who discovered the DNA molecule? Remember the 4 people that are written there. You must know the structural difference between the DNA and RNA. You must know the differences between the DNA, mRNA, tRNA. These are very important. That you must be able to differentiate between DNA replication and protein synthesis, specifically transcription. And then you must know translation and transcription. Learn this off by heart. You see, you must learn them off by heart. That is why I say memorize. Then DNA profiling, which is very important for application questions. So most of the questions that you are going to get, you will be expected to apply. So you learn the process off by heart, but you should also be able to apply it if a question does not ask you to just describe the protein synthesis. And then paternity testing, which involves both parents and the child, and the uses and the interpretations of the word of the DNA profiling. So these are the key points to remember with the first part of the section. Then we also have meiosis. This is 21 marks of paper 2. So again here, we will look at what is it that you need to focus on. So number 1, what is meiosis? Meiosis, it is the type of cell division where the diploid cell undergoes 2 cell divisions to form 4 haploid cells. So we are moving from 2N to N. So from the diploid to haploid. But how many cells are formed at the end? It's 4 haploid cells. And then we need to remember what happens before meiosis. It's sort of like a preparation stage. What does the cell do before it undergoes meiosis? This is where DNA replication takes place. And then know that meiosis again takes place in 2 stages. We have phase 1 and phase 2. We have the first meiotic division and we have the second meiotic division. At the end of the first meiotic division, the result is 2 haploid cells. And then at the end of the second meiotic division, we have 4 haploid cells that are formed, or gametes, because meiosis is a process that results in the formation of the gametes. What are those gametes? The ovum and the sperm cells. Then you must also know why is meiosis important, the biological importance of meiosis. Okay, again here, biological terms are very important. Again, you refer to your list. Those of you who have been good, you go back to your list. So what are the terms that you may come across here? You were told about homologous chromosome, bivalent chromosome. What is a chromosome? What are chromatids? Centromere, centrosomes. What is crossing over? What is a chiasma? What is a karyotype? What is non-disjunction, karyokinesis, and cytokinesis? We've even told you here that you may find these where in question 1.2 in most cases. And remember, we always tell you that in 1.2 you can never compromise the spelling. The spelling is very important in 1.2 because if it changes the meaning of the word, then you don't get the marks. So we said there's a stage in meiosis where the cell prepares itself before it undergoes meiosis, and that stage is what we call interphase. So what happens during interphase? DNA replication. So the DNA makes the exact copies of what? Of itself. So DNA replication occurs. And another thing that takes place in this phase, the unreplicated chromosomes become what? Replicated chromosomes. As you can see here, on the left-hand side, we have homologous chromosome, but at this stage, they are in a format which is called the unreplicated. They appear as singly like this. So they are unreplicated. So this is before DNA replication takes place. And then after DNA replication, this is how they appear. So now we can clearly see the 2 chromatids that are joined by a centromere. And these now represent the replicated chromosome. So those are the things that you must remember. During interphase, DNA replication takes place, unreplicated chromosomes become replicated chromosomes. So with the phase of meiosis, I did tell you that it's 2 stages. We have the first one, this side, which is called the first meiotic division. This is the first, and we have the second one, which is called the second meiotic division. So here the important thing is that when you answer questions, because you will get questions where you are given a diagram, so number one, you must identify what stage is represented here. So what is important when you write here, you cannot just say it is anaphase, telophase, or prophase, because this is not mitosis, this is meiosis. So in your answer, you must be specific whether the phase that is in front of you, is it prophase 1 or prophase 2. So remember these numbers. Or is it metaphase 1 or what? Metaphase 2, because there's 2 stages. First meiotic and second meiotic. So again, with prophase 1, you just need to remember what is important. What takes place in prophase 1 is crossing over. I don't know if you remember this. This takes place in prophase 1, where there's exchange of genetic material between the homologous chromosome. So this brings about variation, which is what my colleague is going to talk about when she does evolution with you. So this genetic variation that we talk about is a result of crossing over in prophase 1. And then what characterizes metaphase 1 is that the chromosomes, they lie along the equator in pairs. So the homologous chromosomes, they lie in pairs. So you can see here the blue and the red. The blue represent one pair, and then the red represent another pair, and then together they are homologous chromosomes. So they lie in pairs and they are attached to the spindle fibers. And then with the anaphase 1, then they are pulled to the opposite pole. So you can see a whole chromosome is pulled to the opposite pole. That is meiosis 1. And then with telophase, then once they reach the pole, the nuclear membrane reappears, the 2 cells are divided into 2, and then we have the 2 cells that are what? That are haploid. And then with phase 2 or stage 2, the 2 cells that were a result of meiosis 1, then they continue again. Again, here we have crossing over taking place in prophase 2, where they exchange genetic material at the points which are called the chiasma, where they touch and they exchange. That is called the chiasma. And then metaphase 2 again is the same as metaphase 1. There's lining along the equator, but here they no longer line in pairs. Now it is single. So in metaphase 1, they were in pairs like this. In metaphase 2, they are now singly. So they form that one line across at the equator. Again, anaphase 2, they separate and they move to the poles. Now, what separates in anaphase 2 are the chromatids. In anaphase 1, we said the whole chromosome. In anaphase 2, chromatids separate, and then at the end, 4 cells are formed. This is just a rough, just a brief description of how meiosis takes place. So remember your blank diagrams on the Mind the Gap. You can take that, try and put in your own notes there when you revise. Check the past question papers if you come across. Remember, questions can be identified. Questions can be identify the phase that has taken place before or the phase that is going to take place after. Questions can be, what do you think will be the number of chromosomes at the end of this division? So you should be able to calculate that. And then with meiosis, you must know non-disjunction and what is the consequence of a non-disjunction. So when we talk about non-disjunction, I know all of you know this, is when we say the chromosomes, when they fail to separate, and when they fail to separate and they all move to this one cell, and then a cell ends up having an extra chromosome. This is what we called non-disjunction. So what causes that? It's when the spindle fibers fail to separate during meiosis. Remember, it can happen in anaphase 1, it can happen in anaphase 2, because those are the 2 stages where the chromosomes or the chromatids separate. So what happens is members of one homologous pair, they fail to separate during anaphase 1 and anaphase 2, And then if there's non-disjunction in pair number 21 in humans, it leads to the formation of the abnormal gamete. So in humans, if there's an abnormal gamete, it is a gamete that has an extra copy, especially away in number 21. And that is what is called non-disjunction. And this results in this disorder, which is called Down syndrome. So Down syndrome, when there's an extra copy on chromosome number 21, not less but extra. So do not confuse this, not less but extra chromosome. So then it will receive an extra, and there will be one that will receive less, but this then will result in Down syndrome. So here we have a diagram that shows that this can happen in anaphase 1, and it can happen where in anaphase 2. So here we have normal separation where the two have gone to two separate cells, but this side it shows non-disjunction where both of them did not separate, but they went on to one cell at the end. And then this is what results in an extra chromosome at the end. Here we have 24 because on number 21 there's an extra, and then this is what we call Down syndrome. Okay, again here, expect questions. You'll be given a karyotype, there will be a karyotype in front of you, and then they will ask you to identify what the problem is. Then you must read from chromosome number 1 up until the last, and then you check what is happening around number 21. Around number 21, that is where if there's an extra, and then you know that then this represents a Down syndrome or non-disjunction. So then that is the end of meiosis. Then the last topic for this first part of today's lesson is genetics and inheritance. This is 48 marks. This is also a very easy topic to pass if you just have paid attention in class when your teachers were teaching you. And if you're also going to pay attention today. Remember, with genetic and inheritance, again, we are looking at biological terms and things that we must know. So what is it that we must know here? Terminology. There's lots and lots of biological terms that come with genetic and inheritance. We have monohybrid crosses. With monohybrid crosses, we are always you know, between homozygous and homozygous, heterozygous, and it depending on whether they are both recessive or there's one dominant or one recessive, different types of monohybrid crosses. And then Mendel's laws as they appear on the exam guideline. And then we have dihybrid crosses where two characteristics are being crossed. And then there's sex determination that you must know. You must know sex-linked inheritance, and you must know what the pedigree diagrams. There's other things that we did not put in here. There's also mutation that you must know when you look at the genetics and inheritance. So those are all important things that you can study under genetics and inheritance. And then what exactly that you must look into? Monohybrid crosses, sex-linked inheritance, pedigree diagrams, not mutation, but genetic engineering. Mutation is in the first part that we did of the DNA and the RNA. Yeah, so here it is genetic engineering. And then you must also know things like paternity testing. And then you understand when we talk about biological terms, the concepts that we are looking into are genetics. In genetics are what is genotype, phenotype, homozygous, heterozygous, allele, genes, a monohybrid cross, a dihybrid cross. Those are all the biological terms that we are referring to, and Mendel's laws. Okay, so the biological terms, remember, these are the ones that we commonly confuse. What is the chromatin? What is the chromosome? What is the gene versus the allele? When do we say the allele is dominant, and when do we say the allele is recessive? What is the phenotype, and what is the genotype? When do we say this is a representation of a homozygous characteristic or a heterozygous characteristic? And when do we say this is a monohybrid cross and this is a dihybrid cross? So we've highlighted this because these ones are the ones that you commonly confuse. So do make sure that you know all these biological terms and you do not confuse them. And then with Mendel's laws, these are on your exam guideline. There's only 3 that we need to know, and these are helpful when we do genetic crosses, because you must understand the laws before you can understand what the genetic cross is. So with the first one, which you call the law of dominance, if you don't know where to find it, it is on page 11 of your exam guideline. It states that two homozygous organisms with contrasting characteristics, when they are crossed, when the two are crossed, all the individuals of the F1 generation will display the dominant trait. So all the individuals, they are heterozygous for a particular characteristic, will have the dominant trait as the phenotype. So we're looking at the two characteristics that are homozygous. It means if you're looking at the red flowers crossed with, let's say, purple flowers, will represent purple flowers with small letter R. So when they say all individuals in the F1 generation will display the dominant, because all of them will be heterozygous, they will look like this, because dominant characteristic is the one that is always displayed. So it means all of them are going to be what? They are going to be red. And then if the heterozygous and the heterozygous is crossed, the offsprings will always display the characteristic of a what? Of the dominant one. And then there is law of dominance. So remember this when you do a genetic cross. And then another one of Mendel's laws is the law of what? Of segregation, found on page 12 of your exam guideline. Here it says an organism possesses how many factors? 2 factors for each characteristic. So all the characteristics that are there, there's 2 factors that control that characteristics. And what happens to those? They separate or they segregate so that each gamete contains only one of these factors. Again here, if you're looking at this one, we have the 2 characteristics. These are the 2 characteristics, 2 capital letter R's. For this individual, they're exactly the same. So what happens during meiosis? These 2 will separate and they will go to each gamete so that each gamete contains only one of what? Of the factors. And then lastly, it's the independent assortment, Mendel's law of independent assortment, which says the various characteristics controlling the different characteristics are separate entities. They do not influence each other. So I might have all these characteristics and as an individual, based on my height, based on the color of the skin, based on the color of my eyes. When meiosis takes place, there's no influence that takes place there. Let's say, for example, the characteristics for being short will not influence how the color of my eyes is going to be, will not influence how the texture of my hair is going to be. This is what is said here. These characteristics are independent of each other and they do not influence one another. And then those are all the laws. Then with the genetic cross, you will be asked, there will be a question where you will be asked to do a genetic cross. Whether it's just a simple monohybrid cross or it is under pedigree diagrams or it is sex-linked related, but there will be a question on how to solve a genetic problem. So here we have been telling you about the format of the genetic cross. When you are given a problem, you're looking at that problem in front of you, and then what other learners do, they just write this layout as it is from the beginning, and then they fill in whatever is missing. So when you do a genetic cross, this is the format, this is the template. Some teachers call it a recipe, depends on what you call it, but this is very important. So here you're looking at the layout or of the genetic diagram. Number 1, you look for what we call the parents in the statement that is given, and you look at the phenotype and the what and the genotype. So what is the phenotype? Remember, the phenotype is the physical characteristic. Are they saying it's brown versus black, or are they saying tall versus short, or is it curly versus straight? That is the phenotype. And then the genotype. Most of the time you will be given the genotype. Once they tell you about curly and straight, they will tell you that, okay, curly is represented by capital letter C, and straight is represented by small letter c. So that will be your genotype. So you write them down there. And then what happens between the genotype and the gametes? A process called meiosis. Please note, when you do this, everything must be in line. You cannot have meiosis that is just after phenotype and then gametes are over there. It makes everything wrong. So you have your meiosis there, and then the gametes, you write your gametes there, then fertilization takes place. Fertilization is always in line with this cross. Sometimes there's a Punnett square there because Punnett square is easier, and we prefer that you use the Punnett square most of the time. And then there's F1 generation. Now, if the question has asked you what will be the genotype or what is the phenotypic ratio if the parents are like this, then you must work out the genotype first before you can tell us what the phenotype is going to look like. And then this is where you will end. So how do you get the marks here? For the fact that you have your P1 and the F1 written out, you get 1 mark. For the fact that you have your meiosis and fertilization written out like that, you get another mark. So that's 2 marks. 3 marks, regardless of what you've written there. Even if what you've written there does not make sense, or you mixed up the letters, or you've mixed up the phenotype and the genotype, you still get the 2 marks. That is why we insist that even if you do not understand the question, when you come to this one, you make sure that you write your format must be correct, because there's 2 marks there. So you check for your phenotype, you check for the genetic makeup, then you check how the alleles separate during meiosis, and then what happens during fertilization. So usually you are looking for a ratio. They will ask you what is the ratio or what is the proportion. If it's the ratio, it is important to write at the end that, okay, the phenotypic ratio, if we're talking about short and tall, it's 3 tall is to 1 short, because the question said what is the phenotypic ratio. So you must show that at the end. Then that's how you get your 6 full marks. So this is very important. Another format, same as that side. The only difference is that with this one, we have used a Punnett square instead of the line. So it's exactly the same thing, but this one is easy because you will put your gametes over there and you will do your cross, and you will have your 6 marks. So remember that by just writing P1 and F1, meiosis and fertilization, you get how many marks? 2 marks. So what are the steps that you must follow? We said you identify the phenotypes of the parents, then you choose the letters. The reason why here we say choose the letters, if the letters were not provided. If they were not provided, you choose. But if they were provided, you use the ones that are given in the statement. Don't come up with your own letters if you're not asked to. And then you identify the genotypes of the parents. Then you show the alleles that are separated through meiosis and then into gametes. And then the next step is to show all the possible genotypes of the F1 generation when after fertilization is what has taken place. And then lastly, you interpret the phenotypes of all the possible genotypes, because you need to work out the genotype first for you to know what the phenotypes are. And then don't forget the types of dominance. These are also common in the papers, where you'll be doing a cross which represents complete dominance or a cross that represents incomplete dominance. So with complete dominant, it is when one allele is completely dominant over the other and the other one is recessive, such that at the end, the effects of the recessive allele are masked. They cannot be seen. It's an example that are shown when we did Mendel's law of dominance. This is what is exactly displayed by complete dominance. So for example here, if we have a homozygous green pod crossed with a homozygous yellow pod, in this case green is what is dominant. So all the genotypes in this generation are going to be green, because green is dominant over yellow, and yellow is not shown because yellow is recessive. There is also what we call incomplete dominance. With incomplete dominance, we are told that neither of the 2 alleles are dominant over the other. So if there's no one that is dominant, this results in an intermediate phenotype. Remember this, you can be asked to explain. So this is a definition of what incomplete dominance is. This is a definition of what complete dominance is. So here neither one of the two alleles of a gene is dominant over the other, resulting in an intermediate phenotype in the heterozygous condition. So here we'll have the white flowers crossed with red flowers. But because there is no dominant between the two, this results in an intermediate one, which is what? Which is pink in this case. So that is incomplete dominance. And then we have the last one, which is codominance. With the codominance here, both alleles are equally dominant. So if they are equally dominant, both of them are going to show themselves because they are both dominant. So we're looking at the cows here, the roan one. The roan color has the mixture of what? Of white and red, because both colors are dominant, so they show themselves. We also see this in the blood types, where a person with blood type A and B, it's an indication of codominance because blood type A is dominant and blood type B is dominant, so both are going to coexist, codominance. So remember the three types of dominance that we're talking about. And then with sex determination, here it is just genetics. We are being told that here, remember, the male is the one that is responsible in determining the sex of the child. Because if you look at the karyotype, this is the female one and this is the male one. Remember, all the chromosomes, number 1 up until number 22, these are what we call the autosomes. All of them, 1 up to 22, we call them the autosomes. And then we have the last pair, which is number 23 in both males and females, the sperm and the ovum. And those last ones are called the The gonosomes. But if you look at the female have 2 X and the male has the X and the and the Y. So the X represent the female, XY represent the male. So what happens during sex determination? A male and the female, so it tells us that each time fertilization takes place, each time a sperm fertilizes an egg, there is always what chance percentage for a male and for a child to be a boy or a child to be a girl. There's always that 50% chance that the child will be a boy or the child will be a girl. It is not known. It does not mean that we're looking at the percentage here, and it does not mean that if, for example, these two are parents and fertilization, reproduction, and they give birth to 4 kids, it does not mean that out of the 4 kids there will be 2 that are males and 2 that are females. It is 1. So it's not actually 50%, but there's 25% chance that it is a male or it is a female. So what happens is the male either donates, we don't use that word. Remember, when gametogenesis takes place, the sperm cells will either have the X or they will have a Y chromosome, whereas the ovum, all the ovum over, they only have 1 chromosome, which is X. And then it depends on the fertilization between the two. So if the ovum that is X is fertilized by a sperm cell that is an X, then the child that will be born there is the female. But if the ovum that is carrying an X is fertilized by a sperm cell that is carrying a Y, then that child will be a boy. So that is why we're talking about the males that are the ones that are able to determine the sex of the child. So that is sex determination. There's always a 50% chance of having a boy and a 50% chance of having a girl. So then with sex-linked determination comes sex-linked disorders. Remember, with the chromosomes carry genes, and then there's different types of genes that are out there. The ones that are found on the sex chromosomes, those genes, we call them the sex-linked genes. So where exactly are they found? They are found on the X chromosome. But here is the problem. The males have X and Y, and the females have X and X. If you look at this diagram, you can see the size of the Y. It is very small. So it does not carry any genes at all because of the size. So most of the genes in the males are found on the X chromosome. Whereas with the females, both X chromosomes are able to carry all these different types of genes. So what happens here is that the fathers can pass the X-linked alleles to their daughters, but not to the sons. Because remember what I said earlier, we said the father, if a mother, an ovum with the X is fertilized with a sperm cell, there is an X, and then the child is a girl. But then if the ovum there is an X is fertilized by a Y, then the child is a what? Is a male. So where does this X and the Y come from? From the father. So the fathers can pass all their X ones to the daughters, but not to where? To the sons. Whereas the mothers can pass both the X's to where? To the sons and daughters. So the sex-linked trait, as we have said, they are found on the gonosomes. Y chromosome is tiny, does not carry any of the genes on it, and these are found where? On the X. So when you represent them, when you do a genetic cross, we usually write them like this. So this one is an example of a hemophilia. So in a female, we will have an X with the superscript capital letter H, depending on whether it is dominant or recessive. If it's recessive, again, this is what we're going to be looking at. But then if it's a male, we'll only represent with one H and Y, which has nothing. Or again here, it can be an X with a capital letter H and a Y, and there's nothing because Y does not carry any chromosomes. There's only two genetic disorders that we need to know. That is hemophilia and color blindness. And then remember we said the father can only pass the X to the daughters, whereas the mother can pass the X to both the daughters and the sons. So again here, if a male without hemophilia is crossed with a mother who has hemophilia, but this mother is a carrier. So being a carrier, it means one of the X will be a capital letter H and the other one will have the small letter H, because this mother is a carrier. So what will be the results? There will be result in a son that has hemophilia, the daughter who is the carrier, the son without, and the daughter who does not carry any hemophilic gene, because in this case the father does not have. So because the father does not have, then it is possible that the daughters will also not have, because the father does not have. But if the father had the hemophilic gene and then we have this one from the mother, then we will have a daughter that is affected. So it depends. Remember, for an individual to be affected, if the disorder is caused by a recessive allele, it means that individual must receive the recessive allele from the mother and the recessive allele from where? From the father. So in this case, it is only the mother that has the recessive allele, but not the father. So the daughters are not going to be affected, but they will be carriers, because remember, the father passes to the daughters only, the mother passes to both. But the sons, there are chances that the sons will be affected because they can receive any of the alleles from the mother. So this is how we differentiate the two. So you will come across questions where it says, let's look at this one. Hemophilia is a genetic disorder caused by a recessive allele on the X chromosome. So a hemophilic female marries a normal male. Explain why all their sons will be hemophilic. So here the female is hemophilic, so both of the H on the X are small letters, and then but the male is not affected, so the male has the capital letter X and Y. So why would the sons be affected? Because remember, the mother can pass on the alleles to both the fathers and to both the sons and daughters. So definitely the sons are going to receive one of the affected X's from the mother and the Y from the father, and they do not have any other X which will mask the effect of the recessive one. So that is why. So let's look at the response, saying all individuals inherit one allele from each other. The Y chromosome was inherited from the father, and the recessive allele was inherited from the mother. Since the mother has 2 recessive allele, a son only needs 1 to be hemophilic, whereas the daughters, we said they need how many? 2 to be hemophilic. So do remember that. Then since the Y does not carry any allele to mask the hemophilic gene, then that is why the sons will all be affected. And then another disorder is color blindness. Then this does not mean that you can only be asked about the 2. So you will get questions where they ask you about a different genetic disorder that you have not studied. But questions like that, they will give you more information as to what causes it, what are the alleles, is it the recessive, the dominant, and even the letters to help you do a genetic cross. Then we also have blood groups that you must know. Remember, with the blood groups, there's 4 types of them. We have 4 blood groups. Human population, there's 4 types of blood groups: A, B, and what? And O. And then these are all the alleles that control these different blood groups. And look at how we write the alleles. There's always a capital letter I if we represent a dominant one, and then the recessive one is represented by a small letter I. And then remember we spoke about the dominance, the type of dominance. With all the blood types, you'll find that they represent complete dominant except for this one. When these 2 come across, come together, we said it's co-dominant because A and B is they are both dominant, so they will show themselves in any phenotypic condition. So this is the one that I'm talking about. Because of time, I'm just going to wrap this one up, and then my colleague is going to take over from me. So with the blood groups, what you need to remember is that we call it the 1, 2, 3, 4 blood group. That the individual has 1 blood group. So if I have blood type A, that is 1. That is number 1. I have 1 type or 1 blood group. But this 1 blood group is controlled by 2 alleles. So it can either be one, both. If it's dominant, it will look like this. If it's heterozygous, then this is it. But it's 2 alleles that control this blood type. And then how many alleles control blood types in general? We have IA, we have IB, and then we have a small letter I. So this is rule number 3. And then in general, at the end, there's 4 blood groups that I've mentioned earlier. So this is blood groups. Unfortunately, because of time, we are not going to be able to do dihybrid crosses, but do remember that dihybrid crosses, they involve 2 characteristics. I'm going to end here, and then my colleague is going to take over, Mrs. Maharaj, and do evolution with you. Thank you very much.
You are responsible for your own life. No matter how old you are responsible for the things that are currently happening in your life right now. Nobody is going to push you to become a better student. Nobody is going to push you to read more, to study more. You are responsible to put in the time necessary so that you can get the results that you want in your life. So today, make sure that you are putting in the work. Make sure that you are responsible in doing everything that you want to do. Because for you to achieve more, you need to put a little bit extra work to what you are doing, and keep on believing and keep on doing, and you will see the success that you want.
Welcome back, grade 12 learners. I'm going to continue with genetics. There's just a little bit that we still need to do, but to actually wrap up what my colleague was talking about, Nhlanhla, please don't forget about the dihybrid crosses. There are marks to be collected there, so please go through it and remember that we are now looking at, it's similar to a monohybrid cross, but now we are looking at 2 different characteristics and not just one. It is also in the slides that you have. You can ask your teachers for it. Okay, so now we're going on to pedigrees and genetic lineages. So what this is about, I'm sure you all recognize this diagram. Okay, it's a very, very important one. You will definitely see it at some point. It's going to be there either in the prelim paper or at the end of the year, but it's a commonly asked question, and it's one of those questions that it really is a skill of how to answer it, how to go about it. It's a skill. It comes with practice, and I'm going to give you some of the tips, remind you about some of the tips of how we can actually go about it. But what are we looking at here? The main thing to remember is that it's the inheritance of characteristics over many generations. The inheritance of characteristics. Okay, so it's like a family tree. It's within a specific family, and they are looking at the generations in that family, what are the characteristics that have been passed down. It is not the same thing as a phylogenetic tree, which we hear from in evolution, which has actually got to do with the evolutionary relationships between species. I'm going to go through that a little bit later, but I just want you to remember that there's a difference. So coming back to the pedigrees, which is now the inheritance of characteristics over generations, this is what it looks like. Okay, so when you see it, you need to know, when you see this, you need to know, what do I need to identify? The first thing is you need to know that all the circles are representing females, and all the circles, yes, are representing females, and all the squares are representing males. Then the next thing we need to look at is that some shapes are shaded and some shapes are unshaded. The shaded ones are the ones that are affected with the characteristic, okay, and then the other ones are not, right? The other thing that I want us to look at is that we need to be able to count the generations. Okay, so every line is a generation. So this will be the first generation, this will be the second generation, and this will be the third generation. Okay, you must be able to count them. Let's look at it a little bit more detail just to remind us of the other things. What do the horizontal and the vertical lines mean? A horizontal line between two individuals, a male and a female, indicates a marriage line. Then the vertical line is the line of descent, and then we also have another horizontal line, but it's looking a little bit different from the previous one. That is the sibling line. Okay, now why is this important? Because the questions they're going to ask you is going to need you to know who is the first generation, who are the parents, who are the offspring, because this is going to help you with determining the inheritance. Okay, before we go further, an important thing to remember, an important thing to understand is that not all pedigree diagrams are showing us sex-linked characteristics. Okay, it can show us, let's just quickly go back to this one. It can show us, okay, actually we can move on to the next one. This is showing us the steps in solving a pedigree diagram. If you look at this, when you are looking here at the phenotype that has been given to us, it's showing us here at the bottom that we have brown eyes and blue eyes. And if you can look at the genotypes, it is indicating to us that this is not sex linked. Okay, they are not sex linked because we do not have X's and Y's. As Nonhlanhla was explained to you earlier, when we have sex linked characteristics, this is when the characteristic of the allele, the allele is carried on the X chromosome. Okay, so then when you have a pedigree diagram that is sex linked, you will be having X's and Y's. But here's an example of one that is not sex linked. It is autosomal characteristic, right? So let's have a look. What do you do first? You study the key, which we spoke about. You identify who's the males and the females. You figured out what is going on. Okay, the squares will be telling you the phenotype, like the previous one that we had over here. Let's just go back. Okay, so when we look at this one over here, what we're looking at here is the phenotype. Okay, these are phenotypes. Let's go forward again. Over here, once again, these are phenotypes. Okay, and then these are the genotypes. Okay, can we see that? So these are the genotypes, sorry. Right, so these are the genotypes. So how do we go about this? You study the key, then you look for the dominant and the recessive characteristics. And then in your pedigree diagram, you are going to fill in the genotype of the individuals with the recessive condition. Why? Because you know, if an individual the phenotype is a recessive condition. For example, blue eyes is recessive. So if the phenotype is a recessive condition, that means that the genotype is homozygous recessive. They're going to have 2 recessive alleles. So that's easy for you to fill out. In this instance, it's going to be small letter b, small letter b. So you will fill out all of them that you can find that is recessive. Then each individual with a recessive condition, what does that basically mean? From what Nonhlanhla was explaining to you, if an individual has 2 recessive alleles, you know that one allele comes from the mother and one allele comes from the father. So if the offspring has 2 recessive alleles, that means that each parent would also have to have a recessive allele. So the next thing you do is you go to the parent generation and you fill in a small letter R for each parent. Okay, you work backwards in this way and you fill in one recessive allele for all the parents, right? If the parent has the dominant characteristic, then you fill in a capital letter for the second letter. Okay, so remember, you must have 2 letters in each block or each circle. Any other individual showing the dominant characteristic is most likely homozygous dominant, which will then have 2 capital letters. Okay. So this is about paternity testing. Just to remind you about the DNA profiling, Nonhlanhla spoke about that you know we can use DNA profiling in order to determine whether the father is the father of a particular child, and we can also use blood groups. But what we must remember is that when we are using blood groups, we can mainly only identify that a specific male is not the father, but we can't guarantee that the male is a father, because what happens with blood groups is that there are many males that can share a specific the same blood group, whereas in DNA profiling and paternity testing it is more exact, because you know that as an offspring, as the child will have 50% of its genes from the mother and 50% from the father. Therefore it is a more accurate indication. Right, quickly, we are not going to go through this question, but we are going to just go through showing you the importance of how we can also have one that is not sex linked. That is why I put this one here particularly, and we're going to do some of the questions, not all of them, but the important thing to notice is that in this instance, they are telling us that this diagram below shows the inheritance of Tay-Sachs, a rare disease which leads to the destruction of neurons. So it is a disease, right? It's inherited as an autosomal disorder controlled by 2 alleles. That's important for you to identify here. And they're giving us here the letters. These 2 should be indicators to you that it is not sex linked. If it is sex linked, if they don't give you hemophilia and they don't give you color blindness, they give you something else. If it is a sex linked disorder, the question will tell you. They will either say it is sex linked, or they will give you they will say something like this, you see, or they'll give you X capital letter T or X small letter t for you to know that it's going to be sex linked. Okay, but we are not having that now, so we are not looking at that. Right, now let's look at our diagram. What did I say? You're first going to fill in quickly. You're going to have a look at everything that's there. You're going to fill in your over here, you're going to have everything over there, and over here you are going to have a look at your questions. Okay, so you will answer them all nicely, but you must be able to indicate. Remember what I said about the first generation, the second generation, and the third generation. Okay, so please go through these. You will also find them on the worksheet that has been given. Right, so just to wrap that up, genetics, really most of it is skills and application. Please make sure that you go through it. Please make sure that you do enough question papers so that you can actually be okay with that section. Right, now comes to evolution. Evolution is another big chunk. It's like a third of paper 2. Right, it's 54 marks, and there's a lot of content here. There's a lot of learning work you really need to study. Okay, 12, there's one thing that I need to emphasize to you is that you really need to study. Okay, so hopefully you have now this whole weekend to study for life sciences. Please make sure that you use your time adequately so that you are able to cover everything. Because look at this, right? This is showing us everything that you need to know on evolution. It's just a bit of a summary from the examination guideline. You need to know the evidence for fossil the evidence for evolution, sources of variation, the Lamarck and Darwinism theories, punctuated equilibrium, artificial selection, speciation, and human evolution. Okay, so it's quite loaded. Right, but we're going to go through it. Right, so these are the evidence of evolution. You should know them: biogeography, genetics, homologous structures, and the fossil record. Right, so what is this basically talking about? When we're talking about biogeography, we are talking about where the species are found and why they are found there. For example, let's look at this picture over here. Here we have flightless birds. You can see that they're found on all different continents. However, they have similar characteristics, and those similar characteristics basically give an indication to us that they shared a common ancestor at some point, and they were also maybe at some point on one larger land mass, which later on divided, because we all know about continental drift, right? Then we've got genetics. Okay, genetics is also about how species are similar to each other based on their genetic makeup. Okay, so the more closely related they are, the more genetic similarities they would have, and the more further, more distant they are, the less similarities they will have. So we look at those to actually see how organisms are more closely related to each other, and if they share more DNA information or genetic information, that means they would share a common ancestor. Then we look at homologous structures. This basically is indicating to us, as you can see here, we have a human, a cat, a whale, and a bat. There are 4 different animals, right? But they all have the similar structure, bone structure, right, in their limbs. Okay, but they have different functions. So this is also an indicator that they could have originated from a common ancestor. Then we have the fossil record. Okay, those are about fossils, and we know that the fossils are found in rocks, okay, in rock layers, and the fossils that are found deeper down into the ground are the older ones. Right, now we look at sources of variation. This is very important. You need to know it, okay, and I've summarized it for you on one page so that it's easier. One page summaries are the way to go. Hopefully you already have them, and all you have to do is just go through them now for Monday. So here we have, there are 2 sources, right? There's the in meiosis and there's reproduction. Okay, so the 2 in meiosis that you need to know is crossing over and random arrangement. Okay, these contribute to variation of the gamete. Crossing over, because in crossing over, as Nonhlanhla explained to you earlier, we have sharing of genetic information, and in random arrangement also there's no specific order that the chromosomes arrange in, they are just arranged, right? Then we have random fertilization for reproduction, random mating, and mutations. Okay, all of these can occur because remember, a mutation is going to be a sudden change in the sequence. So any sudden change is obviously going to affect the organism genotypically, and sometimes then even phenotypically. Okay, and then random fertilization and random mating is also self-explanatory. So I just have a little mnemonic here for you to remember: CRRRM, so that you can help you to remember that C is for crossing over, R's are for random arrangement, random fertilization, random mating, and M is for mutation. Then we have the 2 types of variation that you need to know, which is continuous variation and discontinuous variation. The easiest ways to explain this is by using actually a group of individuals, as you can see over there. In this instance, can you see there are males and females here? But what I want you to also see is that there are differences between them. There's different heights. Okay, we have a variation of different heights. We cannot just say that some of them are tall. We don't just have tall and short. We have various ranges. We have a range of heights. Okay, we also have a range of skin color. We can't just say we have a dark shade and we have a light shade. Okay, we have different shades of color. So that is continuous variation, where we have characteristics that have a range of intermediate phenotypes. Even the hair color we can look at. We can look at yes, hair color as well. Then we come to discontinuous variation. If we look at these same individuals, we can divide them into 2 groups when it comes to gender. We have males and we have females. These are 2 distinct categories. So discontinuous variation is when we divide individuals or anything between two distinct categories, or more than that. For example, look at blood groups. Everybody there is going to have a different blood group, but it's a specific blood group. It's not an intermediate or a blended sort of phenotype, right? And then we have also the fingerprints, which are unique to everybody. Then we have our theories of evolution. There are two, right? You remember them. As soon as I say that, you should know Lamarck and Darwin. And as soon as I say Lamarck, what should you think about? You should think about the two laws, and you should know how to explain them. You should know how to apply them. Please, grade 12s, you need to know this. The law of use and disuse, the inheritance of acquired characteristics. You must know it. If I wake up in the middle of the night and I ask you for the two theories for evolution, it's Lamarck and Darwin. And if I ask you which ones are Darwin's laws, you need to know them. Okay? And you need to also understand what is the difference between an inherited characteristic and an acquired characteristic. This is where learners go wrong. Okay? They confuse the two. Then you also need to know, these are just little guiding questions that you can go through for yourself so that you can see when you faced with the Lamarck question, how do I go about answering? How would Lamarck explain that the giraffes have long necks? Okay, and basically remember what he said. He said that they had shorter necks, and then they carried on using them, frequent use of them to stretch their necks to get the leaves from high up in the trees, and that's how their necks became longer. And then they acquired that characteristic and they passed it down to their offspring. Okay, but how do you apply it to other examples? That's what's over there. Then also, remember, his laws were rejected. Okay, because there's not sufficient evidence to actually prove that the acquired characteristics are inherited by the offspring. Okay, they did not evolve because they wanted to evolve. They cannot just change their DNA. According to Mendel, the genotype determines the phenotype. The genotype determines the phenotype. So it's our genetic makeup, our DNA and our genes that's going to determine our phenotype. Okay. Right, then we have Darwin. Darwin, whenever you wake up in the middle of the night and I ask you what is Darwin's theory, you must tell me natural selection, ma'am. Right, and the natural selection theory, I want you to learn bullet for bullet from the examination guideline. Please, you need to learn it bullet by bullet from the examination guideline. If the question paper is going to ask you to describe the process of natural selection, then you are going to give it bullet for bullet the way it is in the examination guideline. If they ask you for natural selection and they are going to apply, and you need to apply it to a specific scenario, they give you an extract or they tell you about a particular organism, you need to apply your answer to that. So that means that examination guideline bullets that you've learned, you need to make it specific. You cannot just say some have the favorable characteristics, some do not. You need to tell us which characteristic does some have, which characteristics do the others have, which characteristic is then the individual with that specific characteristic will die, the one with the other characteristic will survive and reproduce. We've got an example, don't worry. Okay, so this is about a population of lizards in an island. And what I've done here is I've highlighted for you this where we are making it specific. So you take the examination guideline definition and we're making it specific now. This is about lizards' jaws. Okay, so we first say there's variation in the size of the lizard's jaws. We don't just say there's variation in the population. Okay, it's in the lizard's jaws. Some have small jaws, some have large jaws. So now you need to tell us, what is the variation of the jaws? Be specific. Due to the larger supply of fibrous plants, those with smaller jaws were unable to feed. Those with larger jaws have more food and survive and reproduce. And then the allele for the larger jaws, very important. Learners forget that. It is the allele for the larger jaws, not the characteristic. We are saying the allele for the larger jaws will be passed on to the offspring. Remember, you cannot pass down the characteristic. Okay, remember we said that. Lamarck was rejected. It's the allele, part of the genotype, that is now going to be passed on. The next generation will have a higher proportion of lizards with larger jaws. Okay. So that's very important. Then the third one that we need to know is about punctuated equilibrium. Okay, we need to know the difference between punctuated equilibrium and gradualism. So gradualism was basically what Darwin said. Gradualism is what Darwin said. Okay, so what he said was that over time we are gradually changing and evolving. We have small changes that occur over time, and that we eventually evolve into a different species that way by having these small changes. You can see it happening over here, that we've got this color mouse. You can see the changes that's happening. But in punctuated equilibrium, he's talking about rapid change change occurs and is followed by long periods of no change. Okay, so this was Gould and Eldridge, right? They're the ones who did this in 1971. But what you basically need to understand is that in this one, we have periods of rapid change, and then we have periods where we have little to no change. Okay, so that's what they're saying. They're saying that, like here, for example, we have periods where there's no change, and all of a sudden it becomes white, or it becomes further on, it becomes brown. So we have these rapid changes, not these gradual changes as Darwin would say about the shades that are getting darker or lighter. Then artificial selection. Artificial selection is basically the intentional reproduction of individuals in a population that have desirable traits. Okay, so now with natural selection, basically with natural selection, you understand that nature is a selective force. Okay, but now artificial selection is when humans are the selective force. Okay, humans have a characteristic that they desire. It's a desired characteristic in this instance. Okay, in natural selection, we don't talk about desired characteristics. We talk about favorable characteristics. Okay, but here it is what desirable traits or characteristics that humans would find desirable. For example, you need to know two. Okay, you need to know two of them. So you can either know you need to know a crop species, and you need to know a domesticated animal one. For example, the chickens, we would want bigger chickens. We would want bigger chickens. We'd want fatter chickens. Why? Because it's going to increase our food security. Okay, we are going to want cows that are going to produce more milk. We're going to want to have cattle that's going to produce good meat. Okay, that's what humans want. It's not necessarily beneficial for these organisms, but it's beneficial for humans. That's what you need to understand. The other thing I need you to understand, grade 12s, is that this, you cannot say that this is not necessarily genetic engineering. Okay, it's not happening in a lab. That's what we need to understand. Genetic engineering is like an umbrella, okay? And under there we have biotechnology, we have different things. But this is when you are literally taking one individual and you are breeding them with another individual. That's what I need you to understand there. Okay, you can get lots of questions on that, investigative questions, different types of questions. Just remember to apply yourself. Right, this I want to pause here for a little bit because these 4 terms are very, very important. They are non-negotiable. You need to know them. You need to understand them. Wake you up in the middle of the night, you must be able to tell me. A species is an organism that have same characteristics, capable of random interbreeding to produce fertile offspring. Population are individuals of the same species living in the same area that can randomly interbreed. Speciation is the evolutionary process by which populations evolve to become different species. I want you to notice the differences between species and population. Can you see them there? Okay, so look at the picture. The picture is also going to explain to you quite nicely. Here we have one organism, right? These organisms, this is of a specific species. Okay, when you put them together in a group and they can randomly interbreed with each other, they live in a similar area, it is now called a population. You see, and why am I talking about this? Because this leads us to the next thing, which is called speciation. Once again, from the examination guideline, learn the description off by heart, please, all of the bullets. If they ask you, describe the process of speciation, you will give us the bullets as is from the examination guideline. But if they ask you to explain speciation in relation to a specific they'll give you a scenario, they'll give you an extract, then you need to apply it, okay? So what is speciation? It is the evolutionary process by which populations evolve to become distinct species, okay? And now the one you need to know is speciation through geographic isolation. So what's happening here is that if a population of a single species becomes separated by a geographical barrier, okay, the population of a single species, listen to the words and read the words carefully to make sure you understand, because some learners will learn all of this off by heart so beautifully, and then when it comes into the exam, you confuse when you use population and species, and then you get no marks. Okay? It is a population of a single species gets split. The population itself gets split. Remember, you can't split a species. The population gets split. Remember I told you there's individual organisms of a particular species that live together that can interbreed. That's the population. So now what happens is that this barrier comes in the middle of this population, whether it is a river, it's the mountain range that came up, or something happened, but there is, because of continental drift, the continents moved apart, okay, which caused now sea, right, or water between the two land masses. That is what we're talking about, geographical barriers. So can you see it forces the population to be split? I need you to understand that. So what happens is the population splits into 2 or 3 or 4 or 5 or 6, whatever they give you in a specific scenario. Okay? There is no gene flow between the two populations, because obviously now there's this river. Okay? Since each population may be exposed to different environmental conditions, the selection pressures may be different, and then each population is going to undergo natural selection independently. The individuals become genotypically and phenotypically different, and even if they were to mix again, they will not be able to interbreed. The two populations are now different species. So you need to put it here specifically. Where are you going to be specific, grade 12s? Here you're going to be specific by telling us how many populations there are. You're going to be specific by telling us that if they give you the environmental condition, you need to mention it. You then need to come here to the bottom and you need to name the species that were given to you. Okay? And once again, talk about how many populations we are talking about here. Okay. Also, another thing I need you to understand and remember about this is that there are about 7 marks here. Okay, and sometimes 2 of them may be compulsory marks. You don't need to worry about that. The other place you need to be specific is you need to mention what the barrier is. If the barrier was given to you, you need to be specific to mention where the barrier is. So you see that if you just learn this off by heart and you apply it to the given scenario, you can get your 7 marks. Okay. Then we have mechanisms of reproductive isolation. I want you to know these. I want you to know them specifically. I want you to know them as is. It's from the examination guideline. You say it as is. They love to ask this question. They can give you an example and they're going to ask you what is the mechanism, what is the reproductive mechanism. You need to know them. Okay, please, for example, why I'm saying you need to know them word for word, you must say species specific courtship behavior. You cannot say specific courtship behavior or just courtship behavior. Species specific courtship behavior. Okay, make sure you know them, make sure you know examples of them. Evolution in present times. This is basically about how rapidly reproducing species allow us to study evolution in a short period of time. Okay, these are mainly our microorganisms. Microorganisms, it's insecticides as well. Okay, it's insects. It's showing us how we can actually see evolution occurring, and it's basically the premise of it's basically natural selection. That's what we need you to know from here. It is natural selection. So it's also an application of natural selection that they can give you. They love investigation questions here. Okay, they love to ask investigative questions, but if they're going to ask you to describe what happened to the population of the bacteria, for instance, you are going to apply Darwin's natural selection. Okay, so that is the important takeaway for this part. Now we come to human evolution. Okay, so human evolution, these are the possible questions. Interpretation of phylogenetic trees. You need to know who is the common ancestor, who is extinct, when did the species on the diagram originate, how long did they live for, when did they become extinct. Those are the kind of things that you need to analyze when you're looking at a phylogenetic tree. Similarities between the African apes and humans, so that's what we share. Similarities is what we share with them. Differences is what's different from between us, and specifically you need to know how the skulls differ, bipedalism, foramen magnum, how the spinal cords differ, and the pelvis. Fossil, genetic, and cultural evidence. Know the fossils that were found in South Africa and who found them. And then the out of Africa hypothesis, which is straight from the examination guideline. Please learn all of those bullets off by heart. Right, so here we have a phylogenetic tree. Now, phylogenetic tree shows us evolutionary relationships between species. This is not our family tree as in a pedigree diagram. Yes, it's a family tree within our animal kingdom, okay, and part of our hominids, but it's not like our family tree, like a pedigree diagram is. So don't confuse them. If they give you a diagram that looks like this, remember phylogenetic trees, hopefully you've seen a number of different ones. They can come to you in all different forms. Okay, how are you going to identify it? It's probably going to have a timeline. Okay, it's going to have a timeline to indicate for you something, so you're going to look for that, and then it's going to have these different species. Not one species, it's going to have different species. That's how you know this is a phylogenetic tree. And it can come in any direction. You can read it from this way to that way. It can be from this way up. It can be showing you blocks. It can be showing you lines. It can be showing you anything. Keep your wits about you. It is a skill. Hopefully you've been practicing the skill. Okay? But it's also a commonly asked question. So they like to ask it, so, and it can be quite a few marks, so make sure that you go through a few of them. Okay, and I already mentioned what you need to know from here. Okay, the main things I'm quickly going to mention to you is that you need to identify the common ancestor. Where is the common ancestor? You will find the common ancestor in the past, and 0 is the present. Okay, any line or any rectangle they give you, or any shape, anything they give you that reaches 0, it goes till 0. That is the present. That means that those individuals or those species are still in existence. If the line just stops in the middle, if there's a block box or whatever that just stops in the middle over there, that means that that species became extinct. Okay, then the point where they are diverging from the main line, remember this is the common ancestor, these are the descendants. Okay, any divergence from there is showing us the origin, the new species appearing. Okay, showing us the appearance of a new species. Right, then we have the similarities between Homo sapiens and primates. For this, you need to be able to know you need to know them, right? So for number 1, we have large brains. I'm going to do the actions while I'm going through them, so that you can actually see when you're practicing, you can remind yourself, what are the similarities between us, right? We both have large cranium. Eyes in front, okay? We have freely rotating arms, right? Freely rotating arms. Long upper arms. Elbow joints allow rotation of the forearm. 5 fingers. Opposable thumbs, which will work in opposite direction to the other fingers. Can you see this one's going, thumbs going this way, my fingers are going that way. Then we have flat nails instead of claws or bare fingertips. The 2 mammary glands. Eyes with color vision, or we have eyes with cones, we have color vision, and sexual dimorphism. Okay, so you need to know them. So use your body to be able to do it, okay, so that you can actually remember now, okay, what are the similarities between us. Now the differences. Okay, when we look at the differences, you need to study you need to know the differences like this in a table, but you also need to be able to study the diagrams, because they're probably going to give you a diagram of a skull or a pelvis or a spine, and then you must be able to tell us what the differences are. Okay, so let's first talk about bipedalism. What are the 3 things that are going to indicate to us about bipedalism or quadrupedalism? Okay, and in the skull, it is the foramen magnum. In the spine, it's the shape of the spine, and then the pelvic girdle, it is now where the shorter or wider pelvic girdle. Okay, so now we have for bipedalism, the foramen magnum is in a forward position. For quadrupedalism, it's in a backward position. For the bipedalism in humans, it's S-shaped spine, and in African apes, it is a C-shaped spine. In humans, we have a shorter, wider pelvic girdle, and in African apes, we have a longer and narrower pelvic girdle. Okay, I want to quickly go to the, I'm on the right one. Right, then we have the cranium. So what are we talking about cranium? When we're talking about cranium, we're talking about this part of the skull which houses the brain. This whole head region, the cranium plus the jaws and everything, is the skull. But here we are talking about the cranium, okay? And just to remind you, the foramen magnum is that hole. If you turn the skull upside down, you'll see a hole there where the vertebral column enters. Okay, the spinal cord enters, where the spinal cord enters. That is the foramen magnum. Okay, then we have the palate shape. In humans, we have round jaws, and in African apes, they are rectangular. You can see the actual rectangle box. With dentition, humans have no gaps between the teeth, and dentition for African apes, they have gaps between the teeth, which are called the diastema. Then in humans, we have a non-prognathous face, and in African apes, we've got a prognathous face. So that's this protrusion. Prognathous, we are talking about this protrusion. Then we have a cranial, and we don't have cranial ridges, which is this ridge over here that's found on the cranium. And then we have brow ridges. We also don't have prominent brow ridges, whereas the African apes do have those. And it is for muscle attachment. Okay. Right, so this is just showing you what they look like. You can actually see there the C-shaped spine and all of those. So you need to make sure you know them. Please know those tables off by heart, grade 12. And another thing, you're going to get pictures. Maybe you'll get pictures. So you'll get the skulls, you'll get the spine, you'll get everything. So make sure you go through different ones so that you know the differences and you can identify them when you get them in the exam. Right, the 3 lines of evidence we have fossil evidence. We spoke about it a lot. Where is the fossil found? What's the fossil's name? Who found the fossil? Okay, so when we have similarities in different organisms, when we find different fossils, that's how they identify whether they come from common ancestors or not. Genetic evidence, like we said, they're looking at how similar are humans to African apes with regards to their DNA. Okay, their genetics and mitochondrial DNA. When we're looking at cultural evidence, we are looking at tools, we are looking at artifacts, we are looking at any of those kind of things. And the tools that we have over here in this picture, just to show you, is that you get some more primitive tools and then you get more advanced tools. Okay, so the more primitive tools will actually show that it was used by species that were found longer ago, and then the more recent species will use the more advanced ones there, because as the brain developed and as the cranium became larger, the brains also became more developed and there was greater intelligence. Right, so the fossil evidence, we're not going to go through all of them. You have the tables, but what I want you to remember is that go through these tables because it shows you the species, when it existed, the fossil site, and who it was discovered by. Okay, some African ones you need to know. You need to know Australopithecus africanus, Taung child, Mrs. Blessed Little Foot, and they were found in South Africa, Sterkfontein Caves and Taung, and who found them. You need to know. You need to know that Australopithecus sediba caraba was found in the Malapa cave, near the caves in Malapa, and was found by Elof and Lee Berger. Okay, then you also need to know that Homo naledi, that was the recent discovery in cradle of humankind in South Africa by Lee Berger in 2015. Okay, you need to know them. You also need to know the others, but more specifically, please make sure you know the South African ones. They like to ask them. Right, and then lastly, we have the Out of Africa hypothesis. Here you need to know from the examination guideline, you need to know them off by heart what they are. Basically, what it's saying is that, okay, what we need to understand about the Out of Africa hypothesis is that they are basically saying that humans, the humans' ancestors, okay, remember that we are coming down, our ape-like beings, our hominids, right? They are coming down from a long time ago, and they all originated in Africa, right? Now remember we spoke about how they were bipedal, and then some of them were not, they were bipedal, and when they finally became fully bipedal, they evolved over time. When they could finally become bipedal and walk upright, they actually migrated out of Africa, and that's how we landed up having Homo sapiens all over. Okay, so what you need to understand here is that Ardipithecus fossils were found in Africa only. This is the fossil evidence for the Out of Africa hypothesis. The fossil evidence is saying that the Ardipithecus fossils are found in Africa only. The Australopithecus fossils are found in Africa only. Fossils of Homo habilis are found in Africa only. The oldest fossils of Homo erectus are found in Africa, but the younger fossils of Homo erectus are found also in other parts of the world, which means that Homo erectus was actually the one to move out to other parts of the world. And why do we have evidence for that? Because the fossils of those individuals were found there. The fossils of that specific species was found there. The genetic evidence is mitochondrial DNA, which is the analysis of mutations of the mitochondrial DNA, which trace the oldest female ancestor to East Africa. How amazing is that? Okay, so you need to know that. You need to know that there's fossil evidence for the Out of Africa hypothesis, and you must know that there's genetic evidence. Okay, one other thing that I didn't mention that I quickly want to mention to you is when we are looking at the fossils and we are looking at these different individuals, you need to know sort of how they came. That we had Ardipithecus ramidus, okay? Then we have Australopithecus afarensis. So the first one, that was the most primitive one. Then they developed, okay, into Australopithecus afarensis. Then we've got Australopithecus africanus. Then we have Australopithecus sediba. Then we've got the Homo, right? The Homo genus, which is Homo habilis, Homo erectus, Homo naledi, Homo sapiens. Let's quickly have a look at this scientific names, quickly, right? The first word is the genus. The second word is the species. When you are writing them, the first word, the first letter of the first word must be a capital letter. The first letter of the second word must be a small letter, and you must underline it. Okay. That is scientific names. Please make sure you know this. There are lots of marks to get here. You should get it. Right. Going into the exam room, make sure you have a ruler with numbers. Make sure you have a compass so that you can draw a pie chart. You have a protractor so you can draw a pie chart. You can, you have pencils, you have pens, you have a calculator. Make sure you have 2 pens. Okay? Be prepared when you go into the examination, grade 12s. Really, you need to be prepared. You need to have all of your things. Make sure you study hard. Okay, study everything. Go through at least 5 question papers, and then you should be ready for Monday's paper. Right, so this is a worksheet. I'm just quickly going to show you. It's a worksheet that you can get from your teachers. It is on Pocket School. You can get in the PowerPoint presentation. We also have it here, but you also have a worksheet that you can go through. It's some questions that you can work through on paper too. Okay, so that's all from us for today. Good luck and all the best.